In section 9.10, we talk briefly about cardiac muscle. Cardiac muscle is covered in more detail in chapter 12, but here we do briefly talk about the anatomy and physiology of cardiac muscle cells and compare it and contrast it to both smooth and skeletal muscle. As we look at cardiac muscle cells, we see that they have some similarities to skeletal muscle. They're striated, they have the same proteins found inside the cell, myosin, actin, troponin, and tropomyosin. All of those we saw, of course, at skeletal muscle. And they have well-developed sarcoplasmic reticulum as well as T-tubules, similar to those that we saw in skeletal muscle. Other features of cardiac muscle are more similar to smooth muscle. And so cardiac muscle cells are fairly small, they have a single nucleus, and they do possess gap junctions, just like we saw in the case of smooth muscle. The gap junctions, as you well know, are located in these intercalated discs that we can see here in this photograph and down here in this drawing. And so the gap junctions, of course, provide this connection between adjacent cardiac muscle cells and creating what we refer to as a syncytium, or this collection of cells that works as a single unit in the top half of the heart in the region of the atria and the bottom half of the heart in the region of the ventricles. We are going to begin our discussion of contraction in cardiac muscle cells by looking at figure 9-40 in our text. And so as we look at this figure, we see the entire sequence of events. The very first thing that happens is that we have depolarization of the cardiac cell membrane as a result of the influx of sodium. That, of course, is identical to what we saw in skeletal muscle cells, but the next thing that we see occurring is different. So interestingly, we have an L-type calcium channel, which is a voltage-gated calcium channel located in the plasma membrane of cardiac muscle cells. It opens in response to depolarization because of the influx of sodium, and a relatively small amount of calcium enters the intercellular fluid as a result of that opening. That calcium is not sufficient to bind to troponin, move tropomyosin out of the way, and initiate contraction, but rather what it does is to bind to modified ryanodine receptors that are located in the sarcoplasmic reticulum of the cardiac muscle cell. These are not voltage-gated channels, but these are ligand-gated channels. In fact, they're calcium-gated channels. So this calcium binds to this modified ryanodine receptor. The ryanodine receptor opens. A larger amount of calcium, illustrated by this bold calcium symbol here, exits from the sarcoplasmic reticulum, and calcium levels inside the cardiac muscle cell go up. This, in turn, leads to thin filament activation. In other words, calcium binds to troponin, which is attached to tropomyosin, which moves tropomyosin out of the way, allowing myosin to bind to actin in the sequence of events with which you are already familiar. This leads to cross-bridge cycling and force generation and the overall sliding filament theory of contraction that you know and love at this point. So then what we see is that calcium levels slowly start to decrease inside the cardiac muscle cell. Calcium is pumped back into the sarcoplasmic reticulum, and it's pumped outside the cell. And then eventually, just like we have in the case of skeletal muscle cells and neurons for that matter, we have this exodus of potassium, which leads to repolarization of the cardiac muscle cell. So we see that same basic series of events illustrated in this particular figure from our book. We just want to make note of a couple things that we just discussed. One is that the largest amount of calcium comes from the sarcoplasmic reticulum, not from outside the cell, but the calcium from outside the cell that enters via the L-type channels. The role of that calcium is to bind to the modified ryanodine receptors, which allows them to open and release the calcium from the sarcoplasmic reticulum. Another thing we want to notice is this, that graded contractions are possible and do occur in cardiac muscle cells. And so these contractions are possible because under a normal sequence of events like we just described, then only about 30% of the actin, the myosin binding sites on actin, are exposed. And so through the addition of hormones and autonomic nervous system neurotransmitters, we can increase or decrease the amount of calcium released and therefore modify the strength of contraction of the cardiac muscle cell. The last topic that we want to cover related to cardiac muscle is how L-type channels prevent tetanic contractions in cardiac muscle cells. L stands for long-lasting, and these channels that allow calcium to rush into the cell following depolarization take a long time to close. The end result of this is the following. If we look at these two graphs on the right-hand side of our slide, the top one illustrates the action potential shown in green and the force generation shown in orange that is observed in a skeletal muscle. The bottom graph shows the depolarization shown in green and the force generated shown in orange in a cardiac muscle cell. So because of these L-type channels, we see that the depolarized period is stretched way out, and this means that the refractory period, as illustrated here, is quite long in cardiac muscle cells. So long, in fact, that an additive impact of calcium, like we observed in skeletal muscle when we induce tetanic contractions in lab, is not possible. That's the result of the charge is the result of the following. Let's go over here to the graph on the bottom left-hand side of our slide. So the initial depolarization is the result of the entrance of sodium. That, of course, leads to the entrance of calcium, and so the charge here is the result of both sodium and calcium. And then eventually the drop in charge is the result of both the restoration of sodium and calcium concentrations to their original state and the exodus of potassium, just like we observed in skeletal and in skeletal muscle cells and neurons. So it would be, of course, a disaster if cardiac muscle cells could undergo tetanic contractions, because if they could, then no blood would be able to flow through the heart, or if either chamber, either the atria or ventricles, underwent tetanic contractions, then blood flow would be stopped for that period of time, which would likely be fatal if it was long enough. And so the nature of cardiac muscle cells prevents tetanic contractions through the action of this L-type calcium channel.